You're listening to Season 1 of the QGEM podcast, Parkinson's Unscripted, brought to you by Synaxis.
Hi, everyone. Welcome to this episode of QGEM podcast. My name is Marianna Flores, and I am one of this year's Wet Lab co-leads.
Hi everyone, my name is Zain and I'm also a wet lab lead for this year with QGIM. Um, Today we'll be having our wet lab episode of Parkinson Unscripted, and our guest today is Dr. Marco Prado, who specializes in neuronal communication in neurodegenerative diseases. Dr. Prado, would you like to introduce yourself?
Sure. Thanks for the invitation. My pleasure to be here. My name is Marco Prado, and I'm a professor at the University of Western Ontario in the departments of physiology and pharmacology and anatomy and cell biology, where I also hold a Canada Research Chair in neurochemistry of dementia. And, as you mentioned, I have been working on neurodegenerative diseases for a number of years and interested in how this neurodegenerative diseases spread to the brain, what's the pathogenesis mechanisms, and obviously interested in developing models that we can use to study this disease so we can test for better medications and drugs to treat these diseases.
Okay, amazing. Thank you so much for taking the time to meet with us today. You can just go ahead and get started with the question. So our first question is, a lot of us looking into neurodegenerative diseases from the outside read up on amazing hypotheses surrounding Parkinson's, such as Brack's hypothesis and the brain first models of Lewy body development. However, there still seems to be large amounts of controversy surrounding these models. Could you please give us a small overview of what these models represent and your opinion on the controversy surrounding them?
Yeah. This notion of brain body or body brain, it's really based on one of the pathological hallmarks of Parkinson's disease and other diseases similar to Parkinson's disease, such as Lewy body dementia, Parkinson's disease with dementia, multiple system atrophy. And they actually come from other neurodegenerative diseases. This notion that somehow the pathology that we find in the brain can actually spread from one brain region to another brain region. And in this particular case, also, it's to the point where this pathology may spread from the periphery, right, from the gut, for example, towards the brain through the vagus nerve, through spinal cord, through motor neurons, and reach the places where it would affect, for example, movement. And that is what you see in Parkinson's disease. So, perhaps the most well-studied disease for this is so-called prion diseases, such as mad cow disease, right? So that's where this concept started. This notion that proteins could actually be infectious, right? So Stan Prusner, who won the Nobel Prize for this, and many others have contributed to this as well, have created the term prions to explain these proteinaceous particles that somehow seem to cause disease, cause infection. And so we need to start to go back and understand how the pathogenesis of those diseases happens, right? So you mentioned Lewy body. So a Lewy body, it's a pathological hallmark of this disease. Histologically defined. They're markers. One of the markers of these Lewy bodies are proteins named synuclein. So synuclein, when it gets misfolded and becomes abnormal, it gets also phosphorylated in a specific residue. And because we have really good antibodies against that phosphorylated protein, and we know that in the Lewy bodies that phosphorylated protein accumulates, we use this as a marker, right? So we can identify where synuclein is abnormally phosphorylated at this residue 129. So synuclein is a small protein, about 140 amino acids. And one residue, the 129, gets abnormally phosphorylated on synucleinopathies. So the problem is we don't really know what this abnormally phosphorylated synuclein does, right? But we do know that somehow between a normal conformation of synuclein, right? So synuclein that is, physiologically functioning. And these deposits that we call Lewy bodies, somehow in this continuum, synuclein may become toxic. So the question is this, when we see these deposits, and you can imagine that it's really hard to see this in humans, right? Because, in the beginning, people don't have much of a symptom. So how do you know what this is? And there's no, nowadays, any sort of marker that we can use to see this in vivo with live people, right? So right now, we have science groups working on a technology called post-emission tomography that, for example, in Alzheimer's disease, we can see where plaques in the brain accumulate, right? But for synuclein, we are not quite there yet. So how do we know if it's starting in the gut and the periphery, or if it's starting in the brain? Is there a controversy? I don't think there's actually a lot of controversy in this. I think there are cases where probably you have the disease, this misfolded synuclein that started to accumulate more in the brain. But in other cases, probably a large number of those cases, it may start in the periphery, right? It may start in the gut, for example. And there's good evidence for both. And probably I would say that this is happening because these diseases are not, like, we tend to think of them as one sort of disease, but they're not one disease. They're probably multiple diseases. Same as Alzheimer's disease, right? There's probably not one type of Alzheimer's disease. There are several types of Alzheimer's disease. So answering your question, I mean, we know now that somehow this protein synuclein can be used as a marker of these pathological inclusions that can vary from accumulation to the Lewy body, which is kind of a circle inside the cell that is heavily labeled for this abnormal phosphorylated synuclein, has also mitochondria, has a lot of other stuff in there as well. So we know we can follow this pathology. And now we know from experimental work that this pathology spreads from different parts of the brain to other parts of the brain, from the periphery to the brain, and vice versa. We know this probably occurs in Parkinson's disease and in other types of disease, such as Lewy body dementias, because, for example, if you start accumulating brains of people that died for another reason, right, not for Parkinson's disease, and they die early on, but they did have some Parkinson's sort of Lewy body pathology, you see. And that's what Brack and Brack did, right? They look and they found that there is a spatio-temporal relationship of that accumulation that started in certain places and spread to other places. But we also know from evidence in Parkinson's disease, and this comes from a potential treatment, right? So many years ago, scientists were interested in replacing dopamine neurons in the brain. So in Parkinson's disease, dopamine neurons die. And to do that, they transplanted mesencephalic Parkinson's dopamine neurons to the brains of people that had Parkinson's disease for different reasons, right? So they tried to replace those cells that die in Parkinson's disease by putting embryonic cells that have that phenotype that secrete dopamine. And guess what? Those cells kind of thrive in the brain. If you transplant, we're talking about not a brain transplant, but transplanting cells. And if you put those cells there, they actually, in some patients, they thrive, they improve the way the patients felt and some of the symptoms. What was interesting, though, is that when they went to look, many years afterwards, when people start dying, they could see that some of the new tissue, the neurons that were there, the fetal tissue that was transplanted and implanted in the brain, show the same sort of alpha-synuclein pathology as what you'd see in the tissue from the host, right? So again, why would you see that? This is a completely new tissue from embryonic tissue, so nothing to do with, from somebody from tissues that would not have Parkinson's disease. But you do see the accumulation, and that came, that sort of strengthened the idea that this pathology somehow jumping from place to place, right? So nowadays there is a lot of experimental evidence for that. So there is in vitro experimental evidence that if you put seeds of synuclein in one part of a culture cell, it moves to another part and you can separate them. You can do that in vivo in animal models as well. You can see part of in one part of the brain, and then you find pathology in the other side of the brain afterwards. And if you didn't see it, you wouldn't see anything. So there's really good experimental evidence that this abnormal synuclein or misfolded synuclein may work as a seed, and then it can recruit the whole synuclein, right? And the whole synuclein somehow interacts and changes its conformation and keep changing. It's almost like a self-fulfilling prophecy, right? Like, you recruit more and you change and go forward. And you can do that in vitro, and that's the basis also for some of the cool new potential ways to diagnose if people are going to have, or early on, if they have Parkinson's disease or multiple system atrophy, in a way to try to treat those patients early. So there is ways to do this in vitro, where you get seeds from the spinal fluid, even from the skin, and try to use those seeds to transform normal synuclein in this abnormal form. And you can do that in vitro right now. And then you detect that abnormal form, and so you can sort of predict who may develop those diseases, right? So it's really, in terms of, yes, there are people that may challenge those ideas, but I think there's a lot of us are convinced that this sort of pathology can spread from one part of the body to another part of the body. Does every patient starts in the gut and goes to the brain? We don't know. Probably not, or vice versa, right? Because these diseases are somewhat variable. Does that answer your question?
Yes, absolutely. Yeah, that was very interesting. I didn't know, I wasn't aware about the transplant of the newer tissue and how of those experiments, but that definitely makes sense and definitely is good for comparing experimental evidence and trying to draw conclusions. Thank you. I think we can move on to the next question.
I just had a follow-up question, Dr. Prado. So during your conversation about, this controversy and, this brain first, body first, you brought good points in terms of characterizing how there's many different types of Parkinson's disease, most likely, similar to how there's many different types of Alzheimer's due to, different pathological manifestations of them. So you sort of characterize that alpha-synuclein pathology can either originate in the gut and spread, or originate in the brain and spread retrogradely. But do you believe that there's also a chance of, subset of patients where it spreads simultaneously, or, in most patients would it spread simultaneously from both origin points? Or is it, more so that it starts at one origin and it spreads?
That's a good question. I think the evidence is really that it may start in one particular place and then it spreads to other places. I don't think there's a lot of evidence for happening in several tissues at the same time. However, we should think that, we should take in consideration that, when we say, there are potentially several different diseases, the idea is that probably there are different conformations of synuclein, okay? So, synuclein misfolds, but it may misfold in different ways. And you may have what we call different conformational strains of synuclein, which may give origin to slightly different types of diseases. So we know, for example, that multiple system atrophy and Parkinson's disease or Lewy body dementia, they all have misfolded synuclein. But if you look, people have been able to look at some of that misfolded synuclein with very sophisticated technology, for example, cryo-EM and other technology, and the aggregates, the form of these aggregates are different, right? Suggesting that they're different conformation. So I would say that perhaps that the idea that you have these different conformations may also suggest that spreading might be a little bit, the speed with the spreading might be different between those different conformations as well. we should, I mean, we're talking about this in a way that kind of put all these diseases in the same boat, but they're not. For example, there are some Parkinson's disease that occur because of mutations on synuclein and triplications, right? So there's one extra synuclein gene, so you have extra synuclein, and those can cause Lewy body dementia, Parkinson's disease, right? Mutations as well. So with mutations, for example, which you have a gene mutation, it's conceivable that not, even though not all the tissues change at the same time, that, there might be a more widespread change, as as you're suggesting, that occurring in different tissues more or less at the same time, right? So the problem that we have is that we don't know enough. We don't know enough about the pathogenesis of this disease. We don't know enough about what are the molecular determinants that participate in the conversion of the normal protein to this abnormal form, right? We don't know enough what type of tissues that do this. And, all tissues that have this conversion will be, this protein will be toxic, or just some tissues. So we just don't know enough about this yet, unfortunately. And that's one of the reasons that we don't have disease-modifying therapies yet for synucleinopathies, or even, starting to see some for Alzheimer's disease.
Thank you. That was very insightful. I'm going to, sort of come back to some of the points you mentioned after the second question, because I feel like they relate in. So our second question was that we've talked with, a lot of patients and neurologists about the phenotypic and genetic manifestation of Parkinson's, and we see that the phenotypic manifestation of Parkinson doesn't always do well to mirror its genotypic or biochemical characteristics. And in that sense, what I mean is that, for example, some patients with high amount of alpha-synuclein, right? Sorry. So, for example, like patients with high amount of alpha-synuclein may not have as high of a disease severity as patients with a low amount of alpha-synuclein. And so there's, disparity between alpha-synuclein amount and disease severity. And so, what are your thoughts behind this? And how does it play into the complexity of the pathophysiology behind Parkinsonian diseases that we've been talking about?
Yeah, it has a huge role. So this is not specific for Parkinson's disease or synucleinopathies. In Alzheimer's, you see that as well, right? So you can find people that are, and I'm giving the example of Alzheimer's because that we can actually see in their brains that they have accumulation of their pathological protein, right? Because there are clinical exams that you can use to see that. For example, in Alzheimer's disease, you'd have people that would have their brains full of those amyloid plaques, right? However, they don't have any cognitive impairment, right? I mean, if they have amyloid, they would have a higher risk of developing Alzheimer's, and there's good documentation for that. But they still would be walking around with a large amount of pathology, and they will not have, for example, the symptoms of Alzheimer's disease, right? And so what is important maybe to think is that each one of us is different, right? And we may have some resilience that we don't really know. Part of it could be genetic, right? And there is examples of that. For example, you have mutations of ApoE that increase the risk of developing Alzheimer's disease. You have some mutations of ApoE or APP that actually protect people from having Alzheimer's disease, right? So you can think that probably, or the picture is not yet fully painted, right? So you don't really fully understand what are the genetic, for example, contributions to be, that some people may have more plaques and they don't develop disease, and others that will have plaques or much less plaques and develop disease. I would think with the synuclein it's exactly the same, and with synuclein you have even less knowledge of what we call genetic modifiers. On top of this, there is also what we call the exposome, or the environmental exposure that we are. You probably know this, that for Parkinson's disease, for example, there's an increased risk if people are exposed to insecticides, and if they live in rural areas, etc., right, and have dealt with insecticides or other sort of agents used in farming, right? So that exposome is also an important component, right? So it's likely that I guess you have to think that you probably need more than one component. So misfolding of synuclein is one of the components, but it has to be in that toxic conformation, which, by the way, we have no idea what it is, right? But probably needs to trigger other things. People think about, there is a level of inflammation, for example, that may contribute to these diseases, and that the immune system has a huge role in Parkinson's disease and probably also Alzheimer's disease and other neurodegenerative diseases, right? So, we need to be very careful to study this, because it's very hard to control and know exactly what in your lifetime have you been exposed, right? Unless we have some sort of sensor implanted on people, we really don't know what, other than what they volunteer and said, oh, I've been exposed to that, I've been exposed to that. So it is a very difficult question that we have to probably address in experimental animal models, for example, right? And try to understand, and then maybe translate those findings to humans. So yes, but you're right, not all the time the amount of pathology kind of levels at the same time with the clinical symptoms, but in general it is a good approximation, and you can assume that in most cases that is sort of following the rule that more pathology probably are more advanced in the disease.
So if we were to sort of kind of try to link this back into your work, if what I understanding is right, is that there has to be, it gives a good approximation, but if patients require some sort of predisposing factor caused by environmental stimuli to be able to really increase the rate of misfolding of alpha-synuclein, or increase the effects of misfolded alpha-synuclein on the cell or on the mitochondria or wherever it impacts. Do you see a parallel with this with, the prion disease where you work with, where patients with certain predisposing factors are more susceptible to the effect of, not misfold, infectious prion proteins?
Yeah, I mean, and again, I should emphasize,
Yeah.
it's not just the exposure to the environment, but also
Mhm.
genetic component. And again, people, I mean, there are similarities, particularly for the genetics. It comes to mind a study a few years ago where they used data from 23andMe to look at, a very large number of population for mutations, for example, on the prion protein gene, right? And they even found some mutations that were, that people thought were pathological, and people were not developing disease, right, yet. They also found an interesting thing. They found there's people out there that actually are knockouts for the prion protein gene, right? At least they had one. If I don't make a mistake, they found at least heterozygous people, which means that there might be homozygous people out there going around, which is sort of interesting, because it kind of indicates that if you can't survive without that gene, you could use it to decrease disease, right? In all these diseases, the host protein, right? So the protein that misfolds and is going to be used to amplify that pathology. If you decrease that protein, or if you decrease the expression of that gene, you actually have somewhat of a therapy, right? You delay disease, or you can even prevent the disease as well. So yeah. So there is parallels for prions as well, as you suggest, on this notion that there are modifiers, right? And I would say potentially genetic, and also exposure modifiers as well. And more we studied modifying, there is examples in Alzheimer's disease as well, people that have familial Alzheimer's mutations, and then they found, well, they're not developing the disease in the age they should be developing the disease, and they find out, well, they had another mutation in somewhere else that was protective, right? So there are few examples of that as well. But if you think about average, in average, if you have one of the pathological mutations, you will develop disease, right? Either for familial prion diseases, for alpha-synucleinopathy, or for Alzheimer's disease, you will develop disease. But in some cases, it may be delayed because you have modifiers. It would be important to understand what these modifiers are and can we store them to use as therapeutics, right?
Mhm. Yeah, I think that does really well to, take us into our next question and expand on that a little bit. So, our next question was that there's been, large interest recently on these disease modifying drugs that we spent some time talking about right now, especially for, synucleinopathies and, a lot of other neurodegenerative disorders like Alzheimer's. And so some of these treatments, including monoclonal antibodies, which target, tau or beta amyloid and alpha-synuclein have, seen some success where, where they've been granted two monoclonal antibodies were recently granted FDA approval for use in Alzheimer's. But also, we see that a lot of these disease-modifying therapies that go into these clinical phases, despite reducing the amount of pathological protein, they don't really have an effect on the clinical manifestation of patients. And so, why do you believe that there's this dichotomy in terms of successful and unsuccessful disease-modifying drugs in these neurodegenerative disorders?
Yeah, so again, this is really a good question. We see recently, as you mentioned, that for Alzheimer's disease, two were approved initially, and now there's a third one. And one was actually the first one was sort of abandoned by the company because the second one seemed to be doing better. So it's an interesting conundrum, and there's several reasons for that. Okay, so I'll give you an example first on Alzheimer's and then on synucleinopathies. So, for example, in Alzheimer's disease, probably each one of those monoclonal antibodies, it should be first considered a different drug because they each target something differently. Okay, so the question is, because we don't know what's the toxic conformation, right? So are we targeting the toxic conformation, or targeting something else, or we're targeting one step after the toxic conformation, or 3 or 4 or 5, right? So we don't know that. So that's the first problem. The second problem is a lot of what has been studied with these monoclonal antibodies in vitro or in animal models, right? Indicated that they do what they do. So all the work that was done, for example, in animal models, suggested that these antibodies would be really good to clear amyloid beta from the brain, and they do that in patients, right? So to the point that we're thinking, wow, we may have patients that had plaques and have no plaques anymore. But you're right, you don't see a lot of clinical improvement. You see some, but not a remarkable. And if you look at this, it's sort of interesting. For example, for lecanemab, which is one of the approved drugs, I look at a lot of what has been done in the animal model part of this. And they actually never tested if this antibody improved cognition in these animal models. And when they did a really poor job about it, right? So, and the same thing, there is a cipenimab, for the make a mistake, for synuclein. There was a drug from Roche, and there was some other monoclonal antibodies. I look at very carefully at this. And, a lot of the in vitro work, if it was me, I wouldn't move those things to clinical trials, right? Because yes, it cleared pathology, but there was not really a good indication that was improving, the clinical symptoms that you can detect in the patients. In animal models that will reflect what's happening in patients. So here, there's this one. So there's multiple problems. Okay, so first of all, not all these antibodies probably target the right thing. So that's one thing. Second, you think, because these antibodies have to get to the brain and sort of clear proteins in the brain, and they're not very efficient to get to the brain. So that's another problem. And there's people working on making this better. But a third problem is the sort of preclinical work that has been done with them indicates that they should be doing what they're doing. It's really showing that they can clear plaques and everything, right? But they didn't check whether you'd have the clinical symptoms. So it's a number of different problems that happen that people would have to do better. But obviously, we're learning about all this, right? I mean, there are other problems, and some could argue that the amyloid hypothesis in Alzheimer's disease, for example, is wrong, and it's not that it's not causing any disease, although the genetics says it does. But you could say that in sporadic Alzheimer's disease, it's sort of a component, but not everything, right? So you have different things that contribute for the disease. That's a fourth level of analysis. So all boils down to the fact that we do not understand well this disease yet, but we're trying to treat them, right? So that's the problem. And I guess if I were to advocate for something, it's for more basic understanding of the pathogenesis of this disease, of modeling this disease. For example, a lot of what I'm doing right now is trying to tackle one of these problems. We want to make the preclinical testing in animal models much more robust, more reliable, and potentially predictive, right? And being able to predict if drugs would be successful in clinical trials. So that's something that we're trying to do right now by generating better animal models, better mouse models, maybe getting a gap between mouse and humans with the non-human primate as well, right? Using tests in animal models that are similar to what you can do in humans as well, right? Because, so give an example, in an animal model of Alzheimer's disease, the most common way to look at their behavior cognition is either fear behavior, you give them a shock and see if they remember, or you throw them in a pool full of water and see how long it takes for them to find their way out. Well, you don't shock patients with Alzheimer's disease to figure out if they have memory problems. You don't throw them in a swimming pool and ask them to find their way out, right? The cognitive components are very, very different. So, this comes from this notion that, yes, there's something wrong in the brain. I can detect some sort of something wrong. If I can fix the something wrong, I'll fix it. But this is not really being working out well for the field. So I think we have to take a multi-level approach for this. We have to better understand what is the toxic component of these proteins. For example, for synuclein, what's the toxic component? Can we target that toxic component, right? We need to perhaps do better in the preclinical assessment of this for this disease, understanding better the pathogenesis, what's toxic, and what sort of cognitive dysfunction or motor dysfunction it does cause that is relevant for humans, right? In that sense, we also do a lot of imaging as well, similar to what you do in people with pulsed emission tomography or with MRI, right? So we're not looking at the pathology endpoint, but trying to look at what's toxic. But we also have to do a better job eventually to make sure that some of these drugs can get to the brain and can get better to the brain, right? So it's really sort of multiple approaches that people are taking on this. Everybody's taking, trying to get one of those, at least one of those points. And maybe we also need to understand better the pathogenesis, because maybe it's not just a beta, right? There is also people that think that we really need to treat people, with these antibodies early on, right? Before you have this accumulation of, because when you get a lot of those plaques, for example, the damage is done, right? So it'll be very hard to recover and rescue and go back, even though there's some evidence that you could get some, but it's hard when it passes a threshold. So being able to diagnose this disease is very, very early and try to treat them very, very early might be even so it comes with this notion now that this disease could be defined biologically, not just clinically, right? And you have a set of biomarkers that indicate that, yes, with these diseases, you will with these markers, you develop disease. So we better as well prevent and try to treat it.
Thank you for that. That was definitely really interesting, and it ties in really well with kind of what our end goal is. We're trying to kind of go on the more preventative side as opposed to treatment side, which is what is most widely available on the market currently. So that definitely sheds light on kind of what we're hoping to do as well. And kind of touching on what you were talking about rather like looking at the progression instead of more so, I guess, initial and final states of the toxic fragments and of different neurodegenerative diseases. I wanted to ask you about how we can kind of relate this back to your area of expertise regarding neural communication and how it is affected in these types of diseases. So what kind of like studies regarding molecular and cellular methods are you using to kind of investigate this, or are you how are you kind of what techniques do you use to help gain insights on the processes that are compromised as a result of these diseases?
Yeah, so that's a very good question. So we actually use a very large number of technologies for this, right? So we start from animal models. We try to get as close as possible as the human disease. For example, for convenience and also because of the genetics, mouse models are the key models, right, to start with. So, for example, our approach is to try to get better, to generate better models and use better models. So, for example, for Alzheimer's disease, we generated a mouse model that has 3 different genes that are humanized, similar to what you see in Alzheimer's disease. So, and we're not working with one animal model. We work with 6 Alzheimer's mouse models that are complementary, that have either APOE3 or APOE4, which are major risk factors. They have humanized tau, and they have human APP with or without different mutations, right? And we fully characterize them in several different ways that I'll mention. So animal models is one thing. We just generated fully humanized synuclein mice as well, right? So that animal model part is important because our first generation animal models are transgenic models. Okay, so we know that we need to have the human APP or human synuclein there. So we put a copy of an extra gene in the mouse genome containing that gene, that human alpha synuclein, for example. Okay, perhaps with the mutation that caused Parkinson's disease. So the problem is that to do this, we use what we call promoters to drive the expression of that transgene. And that expression is driven by those promoters, which drives to whatever place those promoters are more active, right? So, and we artificially increase synuclein several-fold instead of what you normally see in those human diseases. So our approach is humanized, but also instead of using transgenic mice, we knock in, so we replace the mouse gene with a human gene. And we put mutations that cause disease as well. So that's one of our approaches, better animal models. We also want to have better understanding of the circuits and what you mentioned, neuronal communication. So we know, for example, that both in Parkinson's disease and in Alzheimer's disease, there's different neurochemical populations that are affected, right? For example, in you mentioned Parkinson's disease, dopamine cells, dopamine neurons are heavily affected. In Alzheimer's disease and even in Parkinson's disease, cholinergic neurons in the basal forebrain are also affected. Excuse me. So it's important for us to be able to measure how these neurochemicals are working in the brain when the animal models develop this disease, because we cannot do that in humans, right? But we can do this in animal models, and we have very sophisticated ways to do this, where we use sensors for these neurotransmitters, for example, or for neuronal activity. They are fluorescent proteins that we can put with a virus in the brain of animal models. And then we can put a fiber optical cable in the brain and record the light from the sensors. So for example, when acetylcholine increases or dopamine increases, there is more fluorescence coming out from the sensors in that particular brain region that you study. But the beauty of this is that you can record those activities, and you can record also neuronal activity by recording intercellular calcium, for example, right? So when you do this, you can actually record very fast at millisecond resolution, And you can do that with the animals doing behaviors, right? They're behaving and they're doing something, trying to remember something. So you see where the neurochemical is affected, or if it's affected in a behavior relevant situation, right? Where they try to remember something, where they're trying to do a motor behavior or so forth, right? So, being able to do this invasive recordings, and in humans, you would do this, you wouldn't do that sort of thing, but you could perhaps use pulsed emission tomography and functional MRI to try to infer about those populations of neurons and circuits as well. So looking at this in terms of recordings, better animal models, another way to do this that we proud ourselves is to use better behaviors. So I have two colleagues that invented new technology to look at behavior in mice. And instead of using the swimming pool that you throw mice in the pool and ask them to figure out where to go, or shock them and ask them to remember where they were when they got shocked, we use touchscreens, basically iPads, where the mouse has to touch the screen and tell us if they remember to see an image or something. And if they do that, and if they get it right, they get some strawberry milkshake as a reward, right? And we can do this in a very comprehensive way, like you do in humans. So they didn't develop just one test for behavior. They developed more than 30 different tests that model what you can do in humans for Alzheimer's disease or for Parkinson's disease, right? And not only that, they have developed tests that are very similar and sometimes identical between mice and humans, right? So we call them this, obviously they're touchscreen tests. And because we have so much of that equipment, and we're the source where this was invented, we can actually test about 800 mice every day in our facilities for their behavior, right? And because everything is automated, all that data goes to a database that we created called mouse bites. So everything goes to this. So we have over 3,000 individual mouse behavior deposit already in the mouse bites. And anybody in the world can use this technology, deposit their data there, or can, once the data is published, we use the principles of open science, so people can go there and collect the data and reanalyze the data, because God knows that maybe we're not always right in our interpretation, so other people could reanalyze and reinterpret, because ultimately what we want is cure those diseases, right? So who cures it is really not what matters to us, but the fact that we may be able to contribute to for the global effort to cure those diseases by improving how we have better animal models, we can do these recordings, we can do a better behavior as a that's sort of high throughput and better, and disseminate those data. And on top of this, we also use very advanced microscopy to look at the brains and look at pathology in the brains. We use a lot of chemistry techniques and molecular biology. But in terms of microscopy, one of the things that we are working now is being able to image a whole mouse brain, right? So one of the problems with microscopy is that light doesn't penetrate in tissues. You probably remember this, right? So if you go to the microscope, you have to use really thin sections. However, there is new technology where we can get rid of what makes the brain opaque, right? We get rid of lipids and we get rid of the stuff that makes the brain opaque. So we make the brain transparent, right? And then we use very fast microscopy and we can image the whole brain with microscopy at the cell detail. So we can see individual cells in very detail. And we couple that with very high power magnetic resonance imaging, MRI. So for example, in people, you do a measure of MRI that has the strength, the field strength is about 1.5 to 3 Tesla, and the max you can use in humans is about 7 Tesla, right? This is the field strength of the MRI. In mice, we just received a 15.2 Tesla MRI. So we can really look, even though the brains are much smaller, we can look in a very, very detailed way, how the brain is working. And obviously, because we do that in humans and you do that in mice, you can try to correlate things between human and mice. So the idea is to really do better preclinical work, more robust, more reproducible, in a way that can impact the treatment for humans. And you can test with these approaches, these treatments, and perhaps predict which of these treatments will work out in humans using these better animal models and better methodology.
Of course, that's really awesome about the new techniques that you guys are using, especially with the whole mouse bites database. I think that's really, really awesome. And I 100% agree with you that the main goal overall is just to help find a prevention for these diseases to limit widespread globally. So I think thank you for sharing that. I
My
think
pleasure.
the last question for today is regarding the treatments that are currently on the market for these neurodegenerative diseases that largely target symptoms as opposed to progression and development of the diseases, as we've been kind of touching upon this entire time. But as such, finding these effective treatments for these diseases remain a significant challenge in the field. Considering the work you've done, are there any specific pharmacological targets that you find have shown promise in your studies, and what about these targets make them stand out as potential avenues for developing new therapies?
Yeah, so I mean, we've been working with a number of different potential targets. I mean, for example, one of the long-standing research questions in the lab is how much neurochemical pathways that are affected contribute to disease. And it looks like, the fact that these neurochemical pathways, for example, the cholinergic system, is dysfunctional, it actually increases pathology and contributes to disease. So that's one thing that is not really curing the disease, but is contributing to the progression of the disease that we're looking at. There's pathways of protein degradation and protein misfolding that we're looking at. And there are two different ways we're looking at this. So one of them is looking at chaperones, so proteins that help and assist other proteins in folding and misfolding. So there's a set of these chaperones, such as heat shock protein 90, heat shock protein 70, and some of those co-chaperones. So we're looking at how those chaperones interact with misfolded proteins. And for example, in synucleinopathies and Lewy body disease, they actually accumulate in the Lewy bodies as well and may be dysfunctional. So we're trying to find ways to correct it. And there is a drug that we're working with a collaborator from New York that targets this pathway. Because of the technology that we've been developing and adopting to do a better preclinical model, we're also collaborating with different companies. And so we have a grant with that company, and this is not a secret because they sent a press release a year ago or more that when we received that grant. So they're working on a vaccine for synucleinopathies. And so they have proprietary technology that can potentially predict the misfolding of synuclein and other proteins. And so they can sort of target that they think they have the right target and the right toxic conformation. And we're testing, I mean, it's still very early, so we cannot say anything yet, but we're testing some of these approaches, some of these vaccines that they are doing in a way to potentially prevent early on the misfolding of proteins and then getting to this toxic conformation that can damage cells. I think we're still working in trying to understand some of the potential positive effects of some of these monoclonal antibodies and some of the drugs that have been recently approved, and see whether early treatments might be the way to go. And we're doing a lot of that. Unfortunately, people haven't done those studies before they went to clinical trials, but we decided that we should do it and better understand the relationship and how well they can clear the brain of pathology and how well they improve cognition. And because we have really sophisticated cognitive tests that are similar between mice and humans, we think that we can better understand whether these drugs, if they were used very early on, they would be less side effects and more effective treatment.
Of course, thank you for that.
My pleasure.
Thank you for your time, Dr. Prado. I think that sort of ends up on all the questions we had. Yeah, I just wanted to thank you for your time, and I think that brings this podcast to an end. Mariana, did you have anything else to add on before we end it off? Any questions?
No questions. Just thank you for meeting with us and sharing your insights.
Oh, no problem. Thanks for the questions and for the opportunity to tell you a little bit about those diseases and the work we do in those diseases.
Amazing. Thank you. Well, we'll see you in the next episode.